Hello, welcome to Biology, the scientific study of life. Antiparallel elongating. As we have noted, the two ends of a DNA strand are different. Notice that. 5 and this one 3. In addition, the two strands of DNA in a double helix are an antiparallel, meaning that they are oriented in opposite direction. Notice that. Clearly, the two new strands formed during DNA replication must also be antiparallel to their template strands. How does the antiparallel structure of the double helix affect replication? DNA polymerases add nucleotides only to the free 3 end of the going DNA strand, never to the 5 end. Notice that. Thus, a new DNA strand can elongate only in the 5 to 3 direction. With this in mind, let's examine a replication fork along one template strand DNA polymers, third abbreviated DNA pol 3 or third one, can synthesize a complementary strand continuously by elongating the new DNA in the mandatory 5 to 3 direction. DNA pol 3 or third simply nestles in the replication fork on that template strand and continuously adds nucleotides to the complementary strand as the fork progresses. The DNA strand made by this mechanism is called the leading strand. To elongate the other new strand of DNA in the mandatory 5 to 3 direction, DNA pol third must walk along the other template strand in the direction away from the replication fork. The DNA strand synthesized in this direction is called the lagging strand, in contrast to the leading strand, which elongates continuously. The lagging strand is synthesized as a series of segments. Once a replication bubble opens for enough, a DNA pol third molecule attaches to the lagging strand's templates and moves away from the replication fork, synthesizing a short segments of DNA. As the double grows, another segments of the lagging strand can be made in a similar way. These segments of the lagging strand are called Okazaki fragments. Notice that. Okazaki fragments. After the Japanese scientists who discovered them, the fragments are about 1,000 to 2,000 nucleotides along the E. coli and 100 to 200 nucleotides along in eukaryotes. Another enzyme, DNA ligase, eventually joins ligates in the sugar phosphate backbones of the Okazaki fragments, forming a signal new DNA strand. So notice that back to Okazaki or Okazaki fragments. Synthesis of leading and lagging strands during DNA replication, DNA polymerase third or DNA pol 3, is closely associated with a protein that encircles the newly synthesized double helix, like notice here. Notice also a double synthesized double helix like a donut. Note that Okazaki fragments are actually much longer than the ones shown here in this figure. We depict only the 5 bases per fragment for simplicity. DNA pol 3 elongates DNA strands only in the 5 to 3. Notice that 5 to 3. 5 to 3. Okazaki fragments, once a new strand, the leading strand can elongate continuously 5 to 3 as the replication fork progresses. Also, the another here, notice that this step, the other new strands, the lagging strand must go in an overall 5 to 3 a direction by addition of short segments Okazaki fragments that grows 5 to 3 numbered here in the order they were made. Notice that 5 to 3. DNA ligase also joins Okazaki fragment by forming a bond between their free ends. This results in a continuous strand. Overall, notice that this one overall direction of replication. Priming DNA synthesis. DNA polymers cannot initiate the synthesis of a polynucleotide. They can only add nucleotides to the free end of an already existing chain that is base paired with the template strand, as we see in this video. The initial nucleotide chain is a short one called a primer. Primers may consist of either DNA or RNA, the other class of nucleic acid, and the initiating the replication of cellular DNA. The primer is a short stretch of RNA with an available free end, and an enzyme called primase can start in an RNA chain from Scotch primase joins RNA nucleotides together one at a time, making a primer complementary to the template strand at the location where initiation of the new DNA strand will occur. Primers are generally 5 to 10 nucleotides long, and third then adds a DNA nucleotide to the free end of the RNA primer and continuous adding DNA nucleotides to the growing DNA strand according to the base pairing rules. Only the primer is required for DNA pol third to begin synthesizing the leading strand for synthesis of the lagging strand. However, each Okazaki fragment must be primed separately. Another DNA polymers 1, DNA pol 1 short term, replaces the RNA nucleotides of the primers with DNA versions, adding them one by one into the free end of the adjacent Okazaki fragment. Fragment 2 in this figure, notice that. But DNA pol 1 cannot join the final nucleotide of this replacement DNA segment to the first DNA nucleotide of the Okazaki fragment, whose primer was just replaced. Fragment 1 in this figure. DNA ligase accomplishes this task, joining the sugar phosphate backbones of all the Okazaki fragments into a continuous DNA strand. Okay, thanks for listening.